Exactly. I was always nice. Most of my patients.
So how many times do you go
Most
to
of
the
my
doctor? I mean, like, you just do checkups?
Well, I have to see a cardiologist now, but he was pretty good, so I don't mind seeing him. And then my PCP like every 6 months. He's moving to the VA, so I'm kind of bummed about that. Because I've had him for 10 years, and he's awesome.
Do they treat you different?
Dr. Boyle. Dr. Wiles or Dr. Boyle.
Will you email those to me?
I sure will.
Where are they located?
They're in Blairsville. Well, one's in Blairsville, one's in Young Paris. And they took care of my sister when she worked up here as a doctor, and she was loved.
Yeah, I've been seeing Dr. Livingston for 10 years. He's awesome, but he's wonderful. But unfortunately, he's going to start working at the VA, which I applaud him for doing that. I worked at the VA for 6 years, but that means I can't see him. My wife could see him, but I can't see him. So.
Oh, send those.
Okay, guys, so here's what we're going to do. We're on Monday. We're going to finish our review for your test Tuesday. We're going to finish up chapter 6, which won't take long. We got like another 5 minutes. And then the genetic stuff. Then I'll go over the lab stuff you need to know. Then we're going to go look at your throat cultures and see if anyone has strep throat. And then if we have some brave volunteers, we're going to do a couple presentations. So, does that sound good? Oh, come on. She's like, yeah, I guess. It will be fun. So where we left off, we were talking about growing bacteria in the lab. And what do you typically need to do that?
The right condition.
Uh-huh.
Osmolarity, good temperature, good nutrients.
Mm-hmm. Yeah, so remember nutrients, temperature, the right amount of oxygen, pH, osmolarity. So those are the big ones you need. And then we talked about differential medium, like, what's an example?
Blood agar.
Blood agar. Why do we call it blood agar?
Because we put 10% sheep blood in it.
5%.
5%?
Yeah, remember, 5% sheep's blood. And it helps you distinguish between different bacteria. That's why it's called a differential medium. So a gram-positive like strep, which is very toxic, will give you beta hemolysis, which is golden yellow, because it destroys all the red blood cells. A less pathogenic variation of a gram-positive like Staph aureus typically will give you alpha hemolysis, which is green or silver or a light brown colony, because it only kills 50% of the red blood cells. And I suspect, honestly, that's what all of you are going to see today in lab. I don't know for a fact, but that would be my guess. This also, basically, if you have a gram-negative, it does nothing. Because gram-negatives, like E. coli, do not release exotoxins. They don't release those digestive enzymes in the environment. So if you put like an E. coli on this, you would just see the streaks from the inoculation loop. And that's known as gamma hemolysis, which is basically no hemolysis. So this is the main one to know. Study the handout I gave you last time, and you will be in good shape. Okay. There's really just one more topic in this chapter, and that's good old binary fission. So you guys tell me, what's binary fission?
And make a copy of the nucleoreduction of bacteria.
Yeah, this is how the majority of prokaryotes reproduce. I'm going to say probably 85-90% of them. And it really is like a watered down mitosis. Basically, the bacteria makes a copy of its nucleoid. It stretches, and then it starts to invaginate the cell wall in the middle, eventually forms a cross wall, and then eventually they pop apart into two daughter cells. And how fast can they do that?
30 minutes.
30 minutes or less. They can do it really fast. And that's why you can grow a whole colony of thousands of bacteria in 48 hours, because they reproduce so quickly. They have what we call exponential growth. And last thing from this chapter, you should know the typical population growth curves. What's that?
When you put it in a medium, how it gets nutrients and grows, and then it stops,
Uh-huh.
and it finally dies.
Yeah. Exactly what they said. So if you put a bacteria in a petri dish, this would be its average lifespan. It would be about 7 days. The first 24 hours or so is what we call the lag phase. You're not going to see a whole lot of growth because the bacteria is kind of scoping out the environment. They haven't quite found the nutrients yet. But then the logarithmic phase, which is the growth phase, this is where they've discovered the nutrients and everyone's eating and going through binary fission. So it's eating, reproducing, and this is where you see the best growth. And it's usually between 48, 72 hours, somewhere in that window. There's lots of nutrients, so all the bacteria are thriving and they're all going through binary fission. Now, after about 72 or maybe 96 hours, 3 to 4 days, they plateau. Meaning the colonies aren't getting any bigger, but they're not getting any smaller either. What's going on?
One
That's
cell
in the
grows and
birth
one
cell.
dies.
Yeah, they basically have gotten rid of 50% of the nutrients. And that's where they plateau and enter what's called the stationary phase. Because they've already eaten 50% of the nutrients, that means for every bacteria cell that's eating and reproducing, another one is starving and dying. So they basically even out. There's really no growth, but no decline. And that lasts about 2 to 3 days, and then eventually, probably about day 6-ish, you start to notice a decline in the size and the number of colonies, and that's known as the death or decline phase. What's going on there?
There's no
They ran out of
Yeah,
food.
pretty much the nutrients are almost completely gone, so you have a lot more cells dying than surviving and reproducing. And that's typically the life cycle of bacteria in petri dish. They don't last forever. They usually have about a week. Okay? Does this make sense? All right, so understand this. You should be in good shape. And that's it for chapter 6. All right, now we're going to get to everyone's favorite chapter, the genetics chapter, right? Yeah. Don't worry, I will make this fun. And we're just going to hit the really important stuff. So what's genetics? Genetics is the study of genes. And the genome is basically all the genes you have in your body. So it's a collection. Genome and genotype are the same thing. It's basically all the genes you have in your body. By the way, what's the definition of a gene?
Specific set of nucleotides within DNA that code for one specific protein.
Yes. Remember this definition. A gene is a specific series of nucleotides. Remember, nucleotides are the building block of DNA. Within DNA that code for one specific protein. That's what they do. So your DNA is made up of millions and millions of nucleotides. You've got millions and millions of genes, and they're all coding for a specific protein. So if this is a part of your DNA, this segment of nucleotides is one gene. It codes for one protein, we'll say like keratin protein. This group of nucleotides up here is a second gene, and it codes for the protein collagen, etc., etc. And you have genes that code for all your enzymes and pretty much every protein you got. So that's the definition of a gene. Definitely know that. And I'm not going to get too nitpicky with the nucleotides, but just remember, what is a nucleotide? Well, it's the building block of DNA. And it's made from a 5-sided sugar, plus a phosphate group, plus one of 4 nitrogen bases. And we say the strands of DNA you know, and a eukaryote is double-stranded in a helix. It's made up of two what we call sugar phosphate backbones for the strands. And they're held in the middle by hydrogen bonding between the nitrogen bases. So you see the strands go in different directions. And each strand is made up of the sugar phosphate component of each individual nucleotide. But since they run in different directions, their nitrogen bases actually face each other in the middle, and they hydrogen bond, and that's what holds your DNA strands together. Now with DNA, how do they bond? Adenine always bonds with thymine. Guanine always bonds with cytosine. Remember that. A always bonds with T. G always bonds with C. So that's absolute. Okay. Gonna jump ahead a little bit. Remember, replication with DNA is semi-conservative. That means when it basically replicates, each daughter DNA is going to have one original strand and one copy of a strand. By the way, which enzyme copies DNA? Polymerase. Write that down if you don't know that. Yeah. The enzyme that actually makes copies of DNA is known as DNA polymerase. First, you have to separate the DNA, usually with DNA gyrase or a similar enzyme. And then once you separate the strands, then DNA polymerase comes in and starts making a copy of each strand. So definitely know that. I won't go into the boring details, but that's basically how you copy DNA. Trust me, this is like a 2-hour lecture by itself. But we have bigger fish to fry. Mainly, you need to know the difference between a genotype and a phenotype. You guys tell me what's the difference between a genotype and a phenotype?
Phenotype is things that are
It's
expressed.
all your genes.
Yes.
Phenotype. Oh, sorry. No, I was like,
No, you guys are good. Yeah, genotype is all the genes you got. So every single gene in your nucleus, if you will, of your DNA is your genotype. Your phenotype are the genes that actually get expressed, and they make up your physical features and your functional characteristics. So we say phenotype, you know, if you're talking about humans, if you're tall or short or brown hair or blonde hair, blue-eyed, green-eyed, that along with your personality traits, that's your phenotype. And that's constructed from the genes that actually got expressed. Now, do most of your genes get expressed? No, maybe 25 to 40% do. So only less than half of your genes actually get expressed and contribute to your phenotype. And when I say genes get expressed, what do I specifically mean by that?
They go through transcription translation.
observe.
That's true, but to get to that point, they have to go through transcription and then translation. So normally if you say a gene has been expressed, that means it went through transcription first, then it went through translation, then it became part of your phenotype. So that's what we mean when a gene gets expressed. So what's transcription? Well, you take a gene in DNA and you transcribe it to a messenger RNA molecule. And that happens in different places depending on what type of organism you're talking about. Where does that happen in a prokaryote?
Cytoplasm.
Cytoplasm. Where does that happen in a eukaryote?
Nucleus.
The nucleus. Definitely remember that, please. And then you take that messenger RNA molecule and it hooks up with the ribosome and it makes a protein. And that always happens where? That always happens in the cytoplasm. So just remember, transcription happens in the cytoplasm for a prokaryote and the nucleus of a eukaryote, but translation, where messenger RNA hooks up with the ribosome and it gets translated to a protein, that only happens in the cytoplasm. And that's for every type of cell. And this is what's known as the central dogma of genetics. Once again, dogma means law. DNA is transcribed to messenger RNA, and then messenger RNA is translated to form a protein. That's the central law of genetics, so definitely remember that. And that's what this is showing. So once again, here's your gene. It's just a specific series of nucleotides. It gets transcribed into a single-stranded messenger RNA molecule, and then that messenger RNA hooks up with the ribosome and the cytoplasm and gets translated by the ribosome into a polypeptide or a protein. And it's that protein that contributes to your phenotype. So this is how you go from genotype to phenotype, and this is how genes get expressed, if you will. Does this make sense?
Mm-hmm.
Yes? Okay. I'm not going to go through all the nitpicky steps of transcription or translation, because I think it's, that's more AMP1 stuff. But I do want you to know the enzyme that is responsible for transcription is RNA polymerase. So RNA polymerase will bind to a certain gene and then will transcribe that gene to make a messenger RNA molecule. So RNA polymerase is the enzyme responsible for transcription. And this is basically what it does. It binds to the gene, it basically goes down the gene, and it's transcribing a messenger RNA molecule as it's heading down the length of the gene, if you will. Now, once it makes that messenger RNA molecule, if you're talking about a eukaryote, that messenger RNA has to leave the nucleus, go into the cytoplasm, and then it's got to hook up with a ribosome. And the ribosome is going to translate that messenger RNA molecule into a protein. Now, one easy way to think about this, what makes up proteins? Amino acids. Remember, that's all a protein is. It's basically a string of amino acids. How does the ribosome know which amino acids to put in what specific order? Codons, yeah, that's basically what messenger RNA is. It's a single-stranded nucleic acid, but it's basically made up of a bunch of codons. A codon is a 3 nitrogen-based sequence that literally codes for an amino acid. And there's 20 different amino acids. So you can see here, like the start codon is adenine, uracil, guanine. Uracil is what we substitute thymine for when it comes to RNA. That codes for the amino acid methionine. And then you have other examples, guanine, cysteine, cytosine, that codes for the amino acid alanine, etc. And that's how the ribosome knows which amino acids to use and what specific order it needs to put them in to make a specific protein. And it's going to keep doing that repeatedly. That's basically what it's doing. It's got the messenger, it's reading the messenger RNA, and then it's adding, multiple amino acids together, forming a protein. How long is it going to do that for?
Until it's a stop.
It's got to hit a stop codon. So there are 3 codons that basically are stop codons until the ribosome, okay, we're done. This protein's done, don't add any more amino acids, just release it into the cytoplasm. And those stop codons are UAA, UAG, or my personal favorite, UGA. Go Dogs. But those are the 3 stop codons. Now, the other thing to remember with translation, where does the ribosome get its amino acids from? Transfer RNA molecules. Do you remember these guys? These transfer RNA molecules are kind of like little helicopters, and they're in the cytoplasm, they're kind of circling around the ribosome, and each one of them carries its own amino acid. And it's just waiting for its turn to be called so it can dive down to the ribosome and drop off its amino acid. How does a transfer RNA molecule know when it's turn to drop off its amino acid? It's going to have an anticodon that's going to be the complementary match to the codon of messenger RNA. Now, what do I mean when I say complementary match?
It pairs.
Yeah, it pairs. Basically, it would bond. It's not identical. So if the anticodon on transfer RNA is adenine, cytosine, cytosine, the codon on messenger RNA would have to be uracil, guanine, guanine. That's what we mean when we say complementary match. So they're not identical, they would just hook up and bond together. So when this codon presents itself in messenger RNA, this transfer RNA molecule goes, oh, it's my turn, it's my turn. It's going to fly on down to the ribosome and temporarily bond with that codon that is the complementary match to its anticodon, and then drop off its amino acid. And then after it drops off the amino acid, the ribosome says thank you very much and kicks it out and moves on to the next codon for the next transfer RNA molecule. And this just happens repeatedly, and you're adding amino acid after amino acid after amino acid until you hit a stop codon. When you hit the stop codon, then literally that's the message to go ahead and stop. So once again, messenger RNA, you have the codons. A codon, just remember that's messenger RNA, and it literally codes for an amino acid. That's the whole purpose. It's a 3 nitrogen-based sequence that codes for a specific amino acid. By comparison, an anticodon is associated with transfer RNA, and that's a complementary match to a specific codon. And the long story short, that tells this particular transfer RNA molecule when it's its turn to come in and drop off its amino acid. So it's very methodical about which transfer RNA molecules come in and which amino acids contribute to building the polypeptide chain. And once again, the ribosome continues to do this until it hits a stop codon. And once it hits a stop codon, then it releases the protein into the cytoplasm. All right, does that make sense? Okay, so that's what I want you to know about transcription and translation. I'm going to mosey on a little bit forward. Are mutations in nature a good thing?
No.
Generally.
No. So here's an important teaching point. Mutations in nature are usually bad. So they're often fatal, or they at least cause a problem. Mutations in the lab, by comparison, are usually beneficial, because usually in the lab we're very specific to what we're trying to do. So a mutation in the lab could be beneficial, but unfortunately, mutations in nature are almost always negative. And sometimes they fail to make a protein, and the organism cannot survive. Now, the main types of mutations we're going to focus on are point mutations. That's where you get one nitrogen-based pair that's affected. You guys tell me, what are the different types of point mutations?
Silent mutation.
Uh-huh. Missense. Yep.
Frame
Nonsense and the frame shift mutations. So a silent mutation means you got the wrong nitrogen base, but just by luck, it made a codon that codes for the same amino acid as the original codon should have. Because if you remember with the codons, sometimes you got 4 or 5 of them that actually code for the exact same amino acid. So basically a silent mutation is, you technically have a mutation because you have the wrong nitrogen base, which means you have the wrong codon. But just by luck, this codon codes for the correct amino acid. So you would actually never know someone had a silent mutation unless you really did extensive genetic testing. Say, oh, by the way, you have a silent mutation, Unfortunately, this is very rare, and it's about the only mutation that's not fatal. By way of comparison, a missense mutation is you've got the wrong nitrogen base, it gives you the wrong codon, and you get the wrong amino acid. However, it's only one incorrect amino acid. It doesn't totally disrupt the entire chain. This can be fatal or it can just cause a disease. This is what causes sickle cell anemia. Basically, in the hemoglobin of a patient with sickle cell anemia, one of the 4 polypeptide chains has a missense mutation. One of the amino acids is wrong. So if their oxygen gets low, the red blood cell changes shape into a sickle. And that can cause blood clots and all types of problems. But interesting, that was thought to be a defense mechanism against malaria. Because if you remember malaria, you have a protozoa that likes to attack red blood cells. It won't attack a sickle-shaped red blood cell. So that was thought to be an evolutionary defense mechanism for folks that lived in an area where malaria was very prevalent. Nowadays, unfortunately, it doesn't really do much, but historically that would make sense. So that's a classic example of a missense mutation. A nonsense mutation is always fatal. What's a nonsense mutation?
Wrong codon for DNA.
Yeah, you unfortunately, because of the wrong nitrogen base, you get a premature stop codon. So if you get a premature stop codon, the protein's never made. And all your proteins are essential. There's no non-essential protein. So basically, if you can't make a protein, that's almost always fatal for the organism. And then frame shift mutations are pretty much always fatal for the organism. What's a frame shift mutation?
One is,
An extra nucleotide.
yeah.
Yeah, you either add an extra nucleotide nitrogen base or you remove one. And the problem with that is not only that codon is going to be incorrect, every codon down the chain is also going to be incorrect. So basically, from the point of that mutation onward, every amino acid is going to be wrong. And you're going to have a non-viable protein, meaning you're going to have a protein that makes no sense and isn't going to work, basically. So that's why frame shift mutations are a big deal. Okay? So just know the difference between the different types of point mutations. Now, what causes mutations? Well, lots of things. Radiation exposure can cause your genes to mutate. Certain chemicals like benzene in the water can cause your genes to mutate. That's what caused cancer. Unfortunately, a lot of the Marines who served at Camp Jejune, actually had a patient when I worked at the VA who had served there in the 80s and had all types of cancer. And he was part of the class action lawsuit where our government finally admitted, yeah, we screwed up. We tried to lie about it for 40 years, but they finally
Is that where
fessed
the men
up.
were getting breast cancer?
Hmm?
Is that where the men were getting breast cancer?
Men were getting breast cancer and all types of cancers that were a little atypical for men. It's because they had benzene in the drinking water at Camp Jejune, and they basically lied about it. So, oh no, there's no correlation with cancer. Tried to blow it off, and finally they lost the lawsuit, and it was proven, yeah, they were lying. So, don't drink benzene, basically. It's bad for you. This is one, too much UV light exposure. This is how you get skin cancer. If you have too much ultraviolet light exposure, it causes what's known as thiamine dimers. What's a thiamine dimer?
It's where two diamonds are beside each other.
Yeah, it's really kind of weird. It's where you just happen to have 2 thiamine nitrogen bases next to each other. And what happens if you get too much ultraviolet light, instead of bonding with the adenines on the other strand, they decide just to bond with each other on the same strand. And that causes a bubble. And what happens is when you try to replicate this DNA, like DNA polymerase comes to that and gets confused. It's like, well, I don't know what to do with this. So it just cuts this part out. He says, okay, we'll just cut that out and jump over here. So you're missing genes. And if that cuts out what's called a proto-oncogene, then you get uncontrolled mitosis, which leads to cancer. So that's why too much UV light's not good for you. It causes thiamine dimers, which can lead to skin cancer.
So, say somebody has more of a, is it just a coincidence that they have more thymine dimers when they get skin cancer more easily?
There's genetic predispositions, so that is true. Okay, typically you see it more in patients that have fairer complexions and such, because they don't have enough melanin to kind of hold off the UV. So, okay. Let me see what else looks important. I think we're almost to the fun stuff. Yes, this is my favorite part of the chapter. Horizontal gene transfer among prokaryotes. Remember, this is something that only happens with prokaryotes. You guys tell me, what's horizontal gene transfer mean?
Transfer of genes within the same generation.
Yeah, you transfer genes within the same generation. So this is where a bacteria cell may mutate its nucleoid and have a gene that's advantageous, so it spreads it to its brothers and sisters in the same colony. So that's what's meant by horizontal gene transfer. It's basically gene transfer within the same generation. So brothers and sisters in the same colony. And there's 3 methods of gene transfer. And you need to know all 3. Transformation, transduction, and the most common is conjugation. I'll say 80% of the time or more it's conjugation. The other 2 happen as
And
well.
this is only for prokaryotes.
Yes, only for prokaryotes. If we could do this, it'd be awesome.
I just want to make sure I had it wrote down right.
Yeah. Only prokaryotes do this. We could do this like I had the gene, I don't get cancer. I could give it to her, she could give it to you, and then none of us would get cancer. Wouldn't that be wonderful? Or diabetes or heart disease. Unfortunately, we can't do that, at least not yet.
Cloning.
Well, don't say that. Unless you played the game BioShock. Anyone play that?
No.
Oh, okay. Remember you had plasmids, you could upgrade your character? I'm probably the only nerd playing this at the moment who understands what that is. Okay, I wasn't the only
You
nerd.
weren't the only one.
But yes. All right. But no, typically this is only in prokaryotes. What's transformation?
Transformation is when they take dead bacteria and harvest the ones parts they want.
Yeah, this is my favorite. I call it the vampire method or the zombie method, whatever floats your boat. But basically, transformation means you have a live bacteria cell that harvests DNA from a dead bacteria and gives itself an upgrade. And it was Dr. Griffin who actually proved this with strep pneumoniae. There was a lethal form of strep pneumoniae that had a capsule around the bacteria cells. And then there was a non-lethal form that did not have a capsule. And then also if you took the lethal form that killed it, it was also harmless. Well, he took the non-lethal strain plus the dead lethal strain, mixed them together, and ended up killing mice. And he's like, well, why did that happen? He did a culture, and he showed the normally non-lethal strains took the DNA or the genes from the dead cells and gave themselves an upgrade. Basically gave themselves capsules, so they became a lethal strain. So that's what happens with transformation. Live bacteria, they can harvest all the genes they want from dead. They can go grocery shopping with a dead bacteria, basically. If they find genes they want, they can upgrade themselves. I hate to sound morbid. Wouldn't that be awesome if we could do that? You know, if you had to, he's resistant to this, I take that. He doesn't get this, I take that. And, give yourself an upgrade. But bacteria.
Gene?
Yeah, exactly. Yeah, be all over that. But that's what bacteria can do. You wonder why bacteria has been around a few billion years longer than us? This is kind of why. They can do stuff like this. What's transduction? This
That's
is
when
kind of
they
interesting.
take the virus
Mhm.
and take, instead of the virus proliferate and takes it into its
Nucleoid.
and takes
Yeah.
the genes it wants.
Yeah, this is very rare, but it does happen. Now, remember, what is a bacteriophage? A bacteriophage is a virus that normally kills bacteria. So what normally happens is it attaches to the cell wall of the bacteria and injects its viral DNA its viral DNA, takes over the nucleoid of the bacteria. The bacteria just becomes a little baby virus factory, and then the baby viruses break out and kill the bacteria. That's normally what happens with the bacteriophage 99% of the time. 1% of the time, you get a super strong bacteria cell. I call this the Chuck Norris or Jason Statham or whoever, Dwayne Johnson, whatever analogy you want to make. This is a super powerful bacteria cell. Bacteriophage tries to infect it, and it's like, oh, thank you very much. I'll take those genes. I'll get myself an upgrade to my DNA. Now run along. You can go now. And that's literally what it does. It takes that viral DNA, but instead of being taken over by it, it takes over the viral DNA and inserts it into its own nucleoid and upgrades its genome. It's very rare, but it does happen. When it does, it's usually a big deal. Usually they want to harvest this bacteria and make multiple copies of it because it can survive a bacteriophage, and that's not easily done. So that is transduction.
Now, if they came across that same bacteriophage, they would have an immunity to it, wouldn't they?
What's that?
If they came across that same bacteriophage.
They would have immunity to it because they've already stolen its DNA once.
Is that the way the COVID vaccine worked? It was an RNA.
It was a RNA virus, but that's not exactly how it worked. It worked because it was messenger RNA for the C protein of COVID. C protein was the protein COVID used to hook on to host cells.
So with the vaccine, though, did they use something similar to that?
No, that's apples and oranges, unfortunately. A good idea, but no, that was not related. What's that?
That'd be good if we could do it.
It would be good, but once we can't,
Viruses are very deadly.
they are. But no, this is the only thing bacteria does, transduction. And even in bacteria, this is rare. You're talking about probably 1 in a million bacteria cells can do this. So, and the last one, which is the most common, is conjugation. We've talked about conjugation a lot. You guys tell me, what is conjugation?
Gets a resistant.
Mm-hmm.
It
It gets a gene that would be beneficial to the colony. Now, normally it's a resistant gene.
This is the one where they become antibiotic resistant.
Yes. But sometimes it can be a fertility gene, meaning it helps them go through binary fission faster or better. So those are your two most common plasmids. It's either a mutated resistant gene where it's resistant to an antibiotic or something, or it's a fertility gene that helps it reproduce faster. So what will happen is the cell that gets I'll just draw over here. We'll say mutates and gets the resistant gene. It wants to pass that resistant gene to all its brothers and sisters in the same colony. So it makes what's called a plasmid. Now, if you remember, the plasmid is only a copy of the resistant gene. It's only a copy of the gene the bacteria wants to pass on. It's not a copy of the whole nucleoid. So the plasmid is always going to be much smaller than the actual nucleoid. Does that make sense? Okay, then what happens?
They send it through.
It's a pilus.
A pilus. Yeah, extends its cell wall.
What is that? So that's just an extension, like same material as their cell wall.
Mm-hmm. They stretch out their cell wall and their plasma membrane and form a pilus, which is basically a bridge. And that hooks up with another bacteria cell. And then they're going to send this plasmid across the pilus to the second bacteria cell like that. Now, what's that second bacteria cell going to do? Yeah, it's going to incorporate that resistant gene into its own nucleoid. Now, this guy is resistant to whatever. But then it doesn't stop there. It decides to pay it forward. Second bacteria makes its own plasmid.
Is they know which one to
They do. Don't get me started.
How do the piluses or pili or pili, how do they connect to the next bacteria?
They're just an extension of the cell wall that hooks up with the next bacteria.
So it just hooks on?
How do they get it back? Did they just
Do they have
re-order back?
consent?
Yep. It's kind of cool, isn't it?
This is alien stuff.
It's like Sandman and Spider-Man,
Yes,
if you know that
exactly.
nerd.
That's-
If you're that nerdy, he
Yeah.
throws his arm and comes back. It's kind of like that.
I swear, this is from aliens.
Well, funny you mention that.
Here we go.
There's some controversy, and I'll just say controversy, over both bacteria and viruses because they're so unusual compared to all eukaryotic life forms if they may be alien in origin. I'm not saying they are. I'm not saying they're not.
But how do they do it? They have their brain, and then how do they know to share this stuff? How do they know?
Almost like they were made by intelligent design.
Exactly. Like
That's all I'm going to say.
like the pyramids.
Exactly. So what happens is this guy knows to send its plasmid to the third bacteria. And now the third bacteria is going to become resistant. And this is just going to keep repeating itself until eventually you have a whole colony of thousands of bacteria that are resistant to the same antibiotic.
And
This is why we don't use penicillin much anymore, except in kids.
does conjugation take about the same time that the other takes?
You mean binary fission?
Yeah.
It's a relatively fast process. I don't know if I would say it's exactly the same length of time, but it's actually a pretty quick process. So the number one method of horizontal gene transfer is conjugation. So I would say 80% of the time, if you're transferring a gene between two prokaryotes, it's going to be through conjugation. However, transformation does happen. So know what transformation is? Transduction happens. So know what that is. They're rare, but they do happen. Okay. Yeah, I could go on for hours about the alien theory, but I won't. But if you're bored, ask me sometime. Okay. That's it for chapter 7. So any questions on that? Okay. Chapter 8 is biotechnology. I like this chapter. I think it's a fun chapter. And once again, we don't do the whole chapter. We're just kind of covering the highlights. So what is biotechnology?
Using microorganisms.
Yeah, to
Make
make
things
practical
help.
products. And we do this by recombinant DNA technology. What's that really mean? Gene splicing. For 50 years, they called this gene splicing. Then they decided that sounded too Frankenstein-ish. So they call it recombinant DNA technology.
So they just made it where you can't hardly say it.
Yeah, exactly. So they say recombinant DNA technology. And folks, oh, that sounds interesting. Okay. It's just gene splicing from 50 years ago. They just give it a prettier name so people wouldn't get freaked out. Why would you want to recombine DNA of prokaryotes?
To eliminate
Eliminate undesirable phenotypes.
Can try to eliminate undesirable phenotypic traits. You can combine the beneficial traits, and this is what they do in agriculture all the time. You know, that's why my wife made super wheat. She cross-fertilized like 20 different types of wheat until she came up with prototype, basically one that won't die. And that's what she got her PhD in from Cornell. I was like, my wife's smarter than me. Oh well.
Tell her I have a raised garden bed that needs some super wheat.
Okay, I will. But all kidding aside, this is honestly what ranchers do all the time. If you have a particular strong strain of cattle, you want to breed it with all the other strains so the offspring have that gene. So you don't have half your cattle die from disease. You lose thousands, if not hundreds of thousands of dollars.
all that, also how they make the American wagon.
American what?
Wagyu. Instead, there's a Japanese.
Oh.
And American. I have a rancher who's making Wagus, which is Wagyu Angus.
Yes.
I thought she meant spaghetti sauce.
Yeah, I was thinking.
Ragu.
They genetically spliced ragu. I was like, what's going on
You
here?
don't have to have the sauce.
Agriculture,
That's why the
they
price
do
went
this
up.
all the time for both plants and animals. If you go to an A&M school like Texas A&M, this is all they do. They have entire science classes dedicated to cross-fertilization for botany or cross-breeding for animals. So you get the strongest offspring, if you will.
For Casey's.
Yeah, so. And then this is the one, creating organisms that synthesize products humans need. And this is the whole recombinant plasmid used to make human insulin and human growth hormone and thyroid hormone. And basically all your synthetic hormones come from this. And it's really been a lifesaver because like with diabetes, with insulin, what did they use before they could figure this out? They used pig insulin. The problem is about a third of the population is deathly allergic to pig insulin. So you were kind of screwed, to be very honest. But when they figured out how to do this in like the early 70s-ish, it saved millions of lives.
Is that why they pretty much stopped using Armour Thyroid?
Mm-hmm. Exactly.
I was on it and it worked so well.
Yeah.
And you couldn't find it anymore.
No, once, I'll be honest, once levothyroxine, which we used to call Synthroid, went generic, it's so cheap now, that kind of took that, because you can make it now for like less than a penny a pill. So now everyone can get that usually pretty cheap. Even without insurance, that's like dirt cheap. Mm-hmm. But historically, they had Armour Thyroid, which you could buy over the counter, but it was super expensive. But yeah, they stopped doing that, so there's no need. Okay, so in order to do recombinant DNA technology, once again, this one's kind of the big picture, where you isolate a gene, you insert it into a plasmid, and then you put that plasmid in a bacteria. We gotta learn a couple things, or review a couple things. First thing you need to know is, what is reverse transcriptase? You guys tell me.
Go back from the mRNA to
Yeah,
it kind of unwinds it.
It's
right?
an enzyme. So it is an enzyme. And with the name reverse transcriptase, it tells you exactly what it is.
Versus transcription.
It reverses transcription. So it will basically hunt down a messenger RNA molecule and then reverses transcription until you get a copy of the original DNA gene. So we put that cDNA. That stands for complementary DNA. I like to call it copy DNA. That's literally what it is. You just made a copy of the original gene. And why is this beneficial?
Because it's easier.
Much easier. I told you about my super nerdy, beautiful mind, former roommate from UGA who was brilliant but crazy. It's the nicest way I can put it.
Most of them are.
Yeah, bingo. He's now a biochemistry professor at Baylor in Texas. Man, he's smart. He makes Sheldon look stupid. I mean, he's like that brilliant, but you can't understand him half the time because he's going on about anyways.
Great lecturer.
Oh yeah, he's a really super nice guy. And if you can understand him, he's brilliant. But even I can't understand half what he's talking about. Okay, that sounds great. I don't remember what that is. Yeah, it's a lot easier to find genes in the cytoplasm with messenger RNA molecules than digging around a nucleus. He's like, you could spend 30 years digging around a nucleus trying to find a certain gene on a nucleotide sequence. Or you could just release reverse transcriptase into the cytoplasm. It'll eventually hunt down and find your gene for you. Because anytime there's hundreds of thousands of messenger RNA molecules floating through the cytoplasm, this guy will hunt down and find the one you want eventually. So it may only take you 6 months to find the gene versus 6 years. So that's why reverse transcriptase is a big deal. Whether it's the gene to make insulin or thyroid hormone or human growth hormone, reverse transcriptase will find it for you. So that's why it's so beneficial. Now, let's say you found that gene you want. Let's say the gene for insulin. Would you want to make copies of it, maybe? So you could send it into multiple bacteria at the same time? Yes. How would you make copies of DNA? PCR. So PCR, let me get there, stands for the polymerase chain reaction. And the reason it's called the polymerase chain reaction is you're using DNA polymerase enzyme to make multiple copies of DNA. And this is how they make copies of DNA. This is used for medicine. It's also, I mentioned, used for law enforcement. This is how they're catching all the bad guys from cold cases. Now they have advanced technology where you can take like the smallest itty bitty tiny bit of DNA, even from 50, 60 years ago, put it in a thermocycler, do PCR, make hundreds of copies, send it to every genome database in the world until you get a match. And now, thanks to Ancestry.com and 23andMe, usually it's like their grandkids or grandnephew or somebody submitted that, they can find a partial match and find, oh wait, this is like a 50% match of somebody who was a prime suspect 50 years ago. And they get a court order to get a DNA test, and it matches, and they go to jail. So that's how they caught the Golden State Killer in California and a few other recent ones. So, like I said, PCR has changed everything. It's changed medicine, it's changed law enforcement. So that's why you guys should know about it. It's
It's
really,
in my PowerPoint.
it is sweet. But this is what you do. It doesn't look like anything fancy. You take a machine called a thermocycler. You take a little test tube of the DNA, you put it in there. And the long story short, what you do is you heat it so the DNA separates. It's called denaturization. You heat it to 94 degrees Celsius, which is about 250 degrees Fahrenheit, guesstimate. And that causes the DNA to separate. And then you load it up with a whole bunch of DNA polymerase enzymes. And those DNA polymerase enzymes just go to work, and they basically start making new copies of each DNA strand. But you run this several times through the thermocycler. So you do it once, you go from one DNA to two DNA molecules. You do it twice, you get 4. You do it 3 times, you get 8. And then 16, 32, 64, 128, etc. So this is how you can make multiple copies of DNA that you want to study. So, very important. Now, if we're talking about recombinant DNA, let me find my magic one. So let's say you used reverse transcriptase. Now you have the gene here, we'll say for insulin. And because you used PCR, you made multiple copies of it. How would you insert that into a plasmid that you could put inside a bacteria cell?
Restriction and something?
Yeah, you need a couple tools. You need what's called restriction enzymes, which I like to call DNA scissors. I know that sounds real scientific, but that's literally what they are. And then you need what's called DNA ligase. What's that?
Glue.
Yeah, that's basically DNA glue. I know that sounds super scientific, but that's honestly what it is. So once you've got the copy of that gene, you take a plasmid and you cut it open using restriction enzymes. So that's what restriction enzymes do. They're enzymes you actually harvest from other bacteria that actually will cut DNA apart at certain sequences. So you use that to cut open the plasmid, and then you insert the gene into the plasmid. The problem is, it's not going to stay unless you do something to kind of glue it in there. And that's where DNA ligase comes in. Once you've got the gene you want inserted into the plasmid, you've got to kind of glue it in there using DNA ligase. When you do that, you have what's called a recombinant plasmid. It's a plasmid where you literally recombine its DNA. And that's where the name comes from. Now you'll insert this recombinant plasmid with the gene that makes, say, human insulin into a bacteria cell. That bacteria cell will incorporate that plasmid in its DNA, and soon you have a whole colony of bacteria that makes human insulin for you. Isn't that cool? But that's literally how they make all synthetic hormones. That's how they make human insulin. That's how they make thyroid hormone. That's how they make growth hormone, estrogen, progesterone, testosterone. Pretty much every human hormone they make is made by this method. Isn't that awesome? I think that's the coolest thing. Now, important teaching point. This recombinant plasmid, because it's introducing a new gene to an organism, what do you technically call that?
Vector.
It's acting as a vector. So it's technically acting as a vector. And in microbiology, a vector means any molecule that introduces new genes to an organism. And 9 times out of 10 in the lab, it's a recombinant plasmid. But any molecule that introduces new genes to an organism is technically called a vector. So that's what recombinant plasmid would be in this case. All right, any questions on this? Does that kind of make sense how this chapter goes together? Okay, good. If you understand this, then you should be in really good shape. One more thing to talk about here. And it's my favorite. Tommy and get a baby woolly mammoth one day.
The dire wolf.
Or a dire wolf, yes.
You can have both.
There we go. Cloning.
Why not?
You guys tell me, how do they do cloning?
Suck out the nucleus of the zygote and put in a new one.
Yep, to be bluntly honest, that's how they do it. They take a zygote, which is a fertilized egg, out of the uterus of a mother, and instead of an oil change, they do a nucleus change. They use what's called a micropipette. These are you have to use like a glass container with those little glove things. You have almost like these microscope goggles so you can see. Basically, you go in and you enucleate the zygote. It means you take out the nucleus. And then you use a separate micropipette containing what you want to clone, and you insert a new nucleus into the zygote. And then once you've inserted the new nucleus into the zygote of whatever you're trying to clone, then you put the zygote back into the uterus of the mother, and the mother gives birth to a clone. That's what Russia's trying to do with the woolly mammoth. They basically had the DNA from fossils of a woolly mammoth. They're going to try to put it into the zygote of an elephant and see if the elephant gives birth to a woolly mammoth baby, which they probably will. But this is how they made Dahlia the sheep, if you remember that. This is how they cloned some rhesus monkeys a couple of years after that. They've actually known how to do this for like 50, 60 years. So they clone animals all the time.
How do we go from not knowing how to do hand washing to cloning?
In a fairly short amount of time.
That's the whole reverse engineering theory, which I will not go into. But my father-in-law believes in that. He's a retired mechanical engineer. But if you start talking to him about that, oh yeah.
How was the hand washing a no-no, but this, yeah, let's do this.
Exactly.
We can't sterilize equipment, but we can take
We
the
can't get
kids
people
out.
to use soap,
What
but
you
we
doing
can
on
get them
Saturday?
cloned.
China is going to try to make dinosaurs, which I think is a horrible idea. But
Yeah, we know how that ends.
yeah. We've seen, what, 6, 7 movies
Yeah.
now, how bad that is. But they're going to take a zygote from a Komodo dragon and put dino DNA in it and see if it gives birth to a dinosaur.
I'm gonna laugh if it was like a ploy that somebody put like in the ground for someone to find one day and it doesn't work.
That would be funny, but yes. So
I just
that's
wonder
cloning.
if the mother animal will have the same instinct. You know, like if this baby's born and, I don't know.
Well, that's the controversy. Often they don't usually let the mother animal raise the offspring,
Oh.
which is kind of odd, but they don't. That's what they did with the dire wolves in Texas.
Yeah.
That's sad.
They basically took a
Take her
timber
baby.
wolf and inserted dire wolf DNA into the zygote, and then once they gave birth, they pretty much are raising the offspring themselves. So,
Rip it from its mama.
Just like Jurassic Park.
yeah, pretty much just like Jurassic Park. Yeah. So, and I forget the logic behind that, but that's typically what they do. So, that's why there's so much controversy with this. And human cloning, obviously, in this country is very illegal. I'm sure they've done it in other countries, let's be bluntly honest. Now, I I don't know if they'll be legal in this country, but they may get to where they can clone organs. And that may be suitable. If you could clone individual organ, like you have a history of liver disease in your family, you could get a clone of your liver and freeze it, and 40 years later, if your liver gives out, you've got one organ to transplant. You don't have to go on a transplant waiting list. And you're not going to reject it because it's genetically identical to your original liver, so you don't even need the rejection drugs. So that will probably happen, and that would be useful. But cloning an entire human being, that's kind of a little too Frankenstein-y for most people, and I agree.
Revelations does say that we can time when people be moved down, so.
Oh, man, we're getting into some serious topics here. Okay. Any questions on cloning?
We can't die is what it's
Okay. Funny, about 3 years ago, I had two sisters in this class that were identical twins. And one of them, on their phone, they had the phone number of her twin as spare parts. So, okay. That's chapter 8. All right. So that's all for your lecture test. I am going to briefly talk about for the lab test. Lab test is going to be pretty straightforward.
Can I, really quickly, I was just looking at the study guide and it mentions photosynthesis. Do we?
Scratch through that.
Yeah, and also, I thought so, chemo, heterotrophs,
Yeah,
and
forget
all.
all that
Okay,
stuff.
thought
It's
so
not
too.
important.
Okay.
So
what
we
I
don't
thought.
need to know, Tim.
If I didn't talk about it, scratch on through.
And operons, same thing.
Yeah,
Okay.
that's the one I told you I dropped about 3 years ago because it takes 45 minutes to explain the operon, and everyone in the class still gets the question wrong. So I'm like, all right, I'm not wasting my time or your time on that. If you're getting a PhD in micro, it'd be important, but you're not, so I don't worry about it.
What about substrate level phosphorylation?
Now, once again, if I did not talk about it,
That's what I thought, but maybe I
it's-
missed it.
So
No.
sometimes it all runs together and I don't remember.
Yes. It's only what I talk about.
Can we scratch the Krebs cycle too?
No, I didn't.
No, I did talk
He
about
actually did
that.
talk about that. I never
Yeah.
heard him say that.
Yeah, I didn't either.
Oh, okay. You keep telling yourself that. Okay. Let me go over what's on the lab test real quick. It's going to be simple. It's going to be the skin culture, the blood agar, which we're going to finish up today, and the parasites. So with skin, obviously know what organisms are common on human skin. So that would be what?
Skin epidermis.
Yeah.
So
You got staph epidermidis, which is the most common, which gives you what type of colonies?
The white.
White colonies on soy agar. You got Staph aureus, which is the second most common, which gives you
The golden colonies.
golden yellow colonies. Which organism causes acne? Propriobacterium diphthoid. So Propriobacterium diphthoid. Don't worry, you won't have to spell that. I'll have it written out for you. That's the microorganism that causes acne, basically. So that's, I won't say that's everything. That's the big picture. Whatever we talked about in lab. The blood agar, strep throat. You should know blood agar, and we've talked about this. I'm going to borrow this real quick. Multiple times. This will be on your lecture test and your lab test. So know what a differential medium is. That's a medium that distinguishes between different bacteria. Know what blood agar is. It's basically soy agar with 5% sheep's blood. And know the difference between beta hemolysis, alpha hemolysis, gamma hemolysis, and know what organisms do which. So strep pyrogenes, which gives you strep throat and scarlet fever and potentially necrotizing fasciitis, that does beta hemolysis, where you have the golden colonies. Staph aureus, strep viridans, strep pneumonia, they're less pathogenic. They give you alpha hemolysis, where the colonies are like green or beige or silver, because you only killed half the red blood cells, not all the red blood cells. And then a gram-negative, like E. coli, you would get gamma hemolysis, which is no hemolysis. That's the big stuff. For the parasites lab, what are the two most common organisms that are parasitic in humans?
Helmet.
Well, protozoa, which Giardia is one,
Helmet.
and helminths and worms. So you should know, let me find my marker. So the most common parasites in humans are either protozoa or helminths. And helminth just means a parasitic worm. Now, protozoa you should be familiar with, probably be like Giardia, which we talked about, Trichomonas, which is basically an STI. Also would have you know Plasmodium. What's Plasmodium cause?
Malaria.
That's the one that causes malaria. So Plasmodium is a protozoa you find in the blood of monkeys. And it doesn't make the monkey sick, but if a mosquito bites the monkey and then bites you, it can transmit that Plasmodium protozoa to you and you'll get malaria. So I'd have you, oh, and I can't forget the last one, sorry. Naegleria fowleri.
Brain eater.
That's the brain-eating amoeba. So I would know these four. For the helminths, you should definitely know, like the tapeworm, which you guys saw an example of that in lab, the bloodworm, and the hookworm. Also, what medication do you use to kill worms?
Praziquantel.
Praziquantel, you should know that. So the medicine that's typically used to kill helminth is called praziquantel. That basically is anti-helminth medication. So long story short, if somebody's got a tapeworm, and you do like a CAT scan or MRI, it's small enough, you can actually give them praziquantel. It will kill the worm and then they'll pass the worm. If the worm's too big, then you have to do surgery to get it out. But praziquantel is kind of the gold standard for killing worms, basically. Lastly, how would you diagnose somebody with parasite infection? You can get a stool sample, that's one way. What's a blood test that helps you?
CBC.
CBC with differential, know that. CBC with differential can help you diagnose parasite infections because you'll see that their eosinophil count is really high. What do eosinophils do?
Well, they help with
Yeah, they're
parasites
the
and
white blood
allergies.
cells that fight parasite infections. They also fight allergic reactions. So if you get a CBC cell blood count with differential, and somebody's got a parasite infection, their eosinophil count should be off the chart. So that would be a good clue, hey, we need to start looking for parasites. So know that, please. If you know that, you should be in great shape. So any questions? No? None? You guys ready to ace the test? All right. Meet me in lab at 11, and we're going to look at your throat cultures and see what you guys grew. And then after that, if we have anyone who wants to do their presentation, we'll come back here and do presentations. So, all right, any questions? All right, I'll see you guys in just a few minutes.
I brought a flash drive. Said my file was too large to email.
That's fine.
I sent you mine in email.
I think I got yours in email, yes.
Yeah. I also have my laptop, and I did come up with a link.